I'd like to thank everybody for coming and finding their way to this end of the massive exhibit hall, and to give us a little bit of time to tell you a little bit about Cellecta technology and then about an interesting application that Dr. Ian Winters is going to talk about in using barcoding technology, which I think really will demonstrate how some of these approaches can be used now that we have the availability of next generation sequencing, the ability to make high quality libraries with oligonucleotides, and how that sort of technology can be applied. But before we get into that, let me just take a few minutes and tell you a little bit about our company, Cellecta. So we're focused really on functional genomics and trying to identify the genes that are driving various biological responses, disease progression, et cetera. And our focus is really to develop technology that allows you to do that in a way that you don't need large-scale automation, you don't need limb systems. You basically can use more or less ordinary laboratory techniques. All of our technology uses standard cell culture or molecular biology techniques. We have a couple of platforms that are focused on doing genetic screening to identify genes that are important for these responses, identifying genes that are activated or deactivated in various conditions, and also looking at the cells and populations of cells or subpopulations of cells that are responsible for the responses or disease progression. And all of them then use ordinary techniques, and then really the output for almost everything is NGS sequencing. So the ability to do large-scale deep sequencing over the last several years has really opened up a lot of possibilities, and the ability to understand what's going on at a much deeper level than we could detect previously using other types of approaches. The company was started in 2007. We're a relatively small operation. We're located in Mountain View, California, which is just south of San Francisco. And we are not a venture-funded company. We started on the basis of SBIR grants, and really we've taken that and put that into developing the platforms and which we offer for services and products in order to be able to carry out these sort of applications. We've had some success over the years. This is just a selected list of some of the references that have used our technology or groups that we've worked with. So the first platform I want to talk a little bit about is DriverMap expression profiling. This is a technique to identify genes that are activated under various conditions, or looking at different samples, seeing which genes are turned on and turned off. It's an alternative to RNA-Seq. It has some advantages over RNA-Seq, but also it's different than RNA-Seq. It's a targeted approach, whereas RNA-Seq is a way to look at really the entire transcriptome. And there's advantages to looking at the entire transcriptome. Obviously, you get much better, more comprehensive impression of what's going on. However, the issue is that you're doing reverse transcription of the whole transcriptome, and it's actually, you're looking at a lot of information and a lot of data, and it can be somewhat time consuming to do. And so depending on what you're looking at, it may be preferable to actually focus on sets of target genes that you're interested in. And we have developed this to target all protein coding genes in the human genome. So we're not talking about looking at 50 genes like you can do with RT-PCR. This is actually looking at 19,000 protein coding genes in the human genome, and it's a single multiplex reaction. So it's a multiplex RT-PCR reaction that you can scale up to 19,000 genes, or you can look at 5,000 genes of interest or 10,000 genes of interest, and I'll show you how it works. It's basically, the key is the sets of primers that we've developed that will target and amplify a specific region of the transcript from 80 to 200 nucleotides for each of the genes in the human genome. So there's 19,000 of these amplicons. We have 38,000 primers, and it's taken a while to develop, but these 38,000 primers work together in a multiplex reaction to specifically target this region of the transcript, one for each gene. And then once the amplification is done, there's a second reaction, PCR reaction, that adds P5 and P7 sequences and indexes. This is so that you can go onto the Illumina sequencer, and then you take that whole set of amplified products, the amplicons, of 19,000 amplicons, sequence them, and the read number you get for each amplicon corresponds then to the expression level of that particular target that it's looking at. And so it's a one-tube reaction. You don't need to do any transfers or any purification in between. You start with total RNA because it's a targeted reaction, so there's no problem with beta globin if you're looking at blood. There's no problem with non-mRNA sequences because you're really only focused on the expression, the gene, the mRNA, sorry, the protein expressing genes. And you can use very low amounts of material because it is a RT-PCR type of reaction. So it's basically qRT-PCR with the NGS readout. So this is just an example of some of the results. This is RNA-Seq at 25 million reads versus DriverMap at 5 million reads. 19,000 amplicons may seem complex, but it's not as complex as RNA-Seq, where you're doing reverse transcription of the whole transcriptome, and you have fragments that you have to align at the end. And so you get actually more sensitivity and deeper reads from a lower number of sequencing reads than you do with RNA-Seq. And the sequencing is basically the same, except as I said, you don't need to go as deep in the sequencing and still get the sensitivity. The preparation can be done in a day. It's first strand synthesis, and then you add the gene-specific primers for the amplification, and then do a second amplification for indexing. And go right to sequencing, and then at the end you have amplicons, and since we know what we're targeting, we have specific reference sequences to align to. So you don't have to do the alignment to the entire to the entire genome. And we actually have, with the kit, we provide a program that will take the FASTQ file and convert that into a spreadsheet so that you have the data right there. So you don't even have to go and do a lot of work in terms of the analysis. I shouldn't say you don't have to do a lot of work in terms of the analysis. You don't have to do a lot of work in terms of getting the FASTQ file into something that you can analyze. But then, of course, the analysis and looking at the expression of the genes is more on the biology side. But you can get right to focusing on that sort of thing and mapping out the pathways of interest. And this is just, I won't go into a lot of detail, but this is just some of the applications that we've used this for with a couple of collaborators. This is looking at total RNA from blood. This is looking at for fibromyalgia markers. But the point is, we have samples here from patients that are affected and patients that are in control, and we were able to identify differences in a set of genes that seem to be differentially expressed in the patients. This is very preliminary, but the point is this is just from 50 nanograms of total RNA directly from blood. And then we also have tumor microenvironment analysis, where we're looking at pre-injected primary tumors and metastatic tumors in the xenograph. And again, this gives you the sensitivity, especially in working with small amounts of material, to be able to do this profiling. And the added feature is that this is in a mouse system. And so we're looking at a human, these are cell lines, the human cell lines that have been injected into the mouse, and that's the primary cells, and then the metastatic tumors. But the metastatic tumors are very tiny, and we took the whole lung and did the profiling. And because it's a targeted system that's specific, you don't have the amplification of the mouse genes in the background. You're really only amplifying the human genes. So we're looking specifically at the human profile. So for a mixed system like that, it actually has some advantages. And then we also have single cell analysis. These are cells that were FACS sorted into a plate that has oligo-DT on each well. And then the cells were lysed, and the cell lysate washed away, leaving the mRNA that is bound to the oligo-DT. The DriverMap primers are added directly to the plate, and then you can go to the indexing, and then we can do the sequencing. And so you can see here that we got very clean profiles between the 293 HEK cells and the JERCAT cells, and so it was very easy to differentiate them. And this is a single cell approach, basically, but it's a single cell approach with where you have to keep each of the samples separate. We are actually working on developing a single cell approach now, where the indexing is done at the first step of the reaction with the RT, so that you can combine all the samples and be able to then run the reaction in bulk, the actual PCR. We offer this as a off-the-shelf product. We also have a service where we can do the profiling, and we can also make customized assays. For human, it's no problem because we have all the primers. For mouse, we have primers up to about 5,000 target genes at this point. For other organisms, we'd have to develop them, and it would be a much more involved project. But we have now, done this a few times, and so we have the capability of doing that if there's some interest. So I'll just talk a little bit also about the CRISPR technology and the genetic screening that we've done, and this is really what the company was founded around. The company started in 2007, as I mentioned, and at that time we were focused really on shRNA, RNAi, and it was pooled RNAi technology, pooled RNAi screens, and then rapidly with the publications in 2012, 2013, for the screening, 2013, we started to get involved in looking at shRNA and doing pooled shRNA libraries. And these are really the only two technologies that you can use to do this type of screening, because they're both effective for knocking out genes or disrupting various genes. And there are other technologies that are effective also, but you can design just using information in the databases to basically any set of genes, just using sequence information, these effective reagents. So you can design very easily for 80,000 shRNA that target all 19,000 protein coding genes, for example, which is a genome-wide library. And the approach to make libraries with either shRNA or sgRNA is the same. We actually work with Agilent to make the oligos for oligosynthesis. So we design several thousand oligos that are targeting whatever set of genes are of interest. These are then cloned into a lentiviral construct, and we use lentivirus because it can be packaged, and so it's packaged as lentivirus, and it's effective then to transduce almost any type of cell, but it also transduces in a very even distribution. So when you do the transduction, you have to make sure that you're introducing the library in a very representative manner, and so you get approximately one sgRNA or one effector per cell. And so by adjusting the ratio of the virus to the cells, you can do a low MOI and you can get a nice distribution, and then you just select the cells that have the virus, and then you can move on with the screen. And the screening is done any number of ways depending on what you're looking at. The most popular screen, especially with this group, is really to identify essential genes. And so in that case, you allow the cells to grow for a period of time, and then isolate the genomic DNA, sequence that by next generation sequencing, and then take a look at the number of reads for each one of the guides in the library, in the original library compared to the experimental population after growth for a couple of weeks. And so you'll see that some of them are depleted, most of them are on the diagonal and have not really changed in terms of their representation. But some of them are depleted, and those that are depleted then are lethal to the cells, and they're lethal to the cells presumably because they're targeting genes that are essential. So that's one way of doing the screen. You can actually hit it. That's a very popular way also to try and identify genes that are important for resistance, because those are selectively essential under certain conditions with the drug as opposed to without. You can hit it with a large dose of a drug or factor and try and kill the cells, and then identify genes that are important for that response, that apoptotic response, say, or something like that. You can also vary this by using FACS type of screening to do some sorts of selection. So there's many ways you can use this approach. We focused really on the screening, but I just want to point out, without going into a lot of detail, we've done some work. This is the original sgRNA that was developed by the Dudna and Charpentier labs, and this is derived from bacteria. In bacteria, these are two different strands, but what they did is actually combine them to make what's called a single guide molecule. And so this is actually the structure that's been used by most groups, even up till now. However, there are some differences between this and the original bacteria. There's several bases that were omitted when this was made into a single guide molecule that there's some evidence if you add it back, you get better Cas binding. And also there's a row of U's here which function as a stop signal for the construct that's used in eukaryotes, in mammalian cells. So it's not a problem obviously in bacteria, but in mammalian cells it is. So we tested a couple different variations of the sgRNA that had a AT swap here, or had a additional bases from the bacterial sequence that are added back into the single guide molecule to look and see how that affected screening. And the reason we wanted to do this is because if this gave us better results in terms of a screen, it's something that can be applied across the board to all sgRNA in the library. And so we made 4 different libraries, and just to cut to the end, if you look at what we call the wild type, or the standard sgRNA here in blue, versus the one that has both the helix extension, the hybridization extension, and the AT inversion, which we call HEAT, in yellow, you can see that consistently the addition of both mutations gave us stronger signals in the screen. So not only do you get more effective Cas binding, which was shown already, but the cutting seems to be faster, and actually that translates to a better signal in screening. And then just to point out that there are some innovations of the CRISPR system that you're probably aware of, where a couple of nucleotides have been used to deactivate the Cas9 protein. And then what this gives you is sort of a targeting signal now. So now you have a guide that you can use to target this Cas9 protein really anywhere in the genome. And then you can add functions. So you can join this to some other protein, either by fusing or by other mechanisms. And say, for example, add a transcription activator or a couple transcription activators and use that to, I should have had this upside down, use that to activate or upregulate expression. Or in the case of CRAB, which is a repressor, you can add that, and then that will deactivate. So these are the CRISPR-I and CRISPR-A systems. And there are other types of variations of CRISPR coming out. So these variations of CRISPR, I think, will be a platform that will be continued to develop for many years to come. But in terms of CRISPR-A and CRISPR-I, we did some work on that. We have an activator complex that we use, but we also have a number of libraries off the shelf, and we can again make custom libraries for mouse and human CRISPR-A and CRISPR-I. And then also some ancillary products such as these CRISPR test kits, which are fact-based assays to identify the activity of the Cas9 in their cells, or the CRISPR, the dCas9 activator or dCas9 repressor when you make the cells before you run a screen. And some other products too. So as I mentioned, if you have any questions about this, or if you're interested in doing these type of screens, please stop by our booth and we can talk a little bit more about that. And then I want to talk a little bit about barcode libraries at this point. So as I mentioned, we've been doing for many years making oligo libraries with first for shRNA, then with CRISPR, and we can synthesize thousands of oligonucleotides and clone these in mass into a lentiviral construct. So you can also design the oligos that just have a unique sequence, and you can design them using the informatics to make sure, for example, that there are a couple different bases to go from one sequence to another. So if you're reading them on a sequencer, you have to have a few different, you can uniquely identify each oligo. So that'll give you a library with a complexity of, say, 100,000 or 10,000, 50,000 or 100,000. But you can, using a little bit of cloning tricks, you can basically take a couple of pools of these and then clone these into a construct. And what happens then is now you have sort of, if you have 10,000 and 10,000 different oligo pools on each side, 10,000 times 10,000 is 100 million. So now you can make a very complex library. And since you've defined these very carefully, these are well-defined, clear oligos that you're targeting. So again, you have reference sequences. So this isn't random synthesis. And then this is a lentiviral library. It can be packaged and introduced into cells. And now if you introduce this at a low MOI into cells, you can label each one of your cells with a particular barcode. And then that gives you the ability to then do some studies where, for example, you can label a population of cells. You can grow them in vitro or grow them in a mouse. Go out several weeks, allow them to grow. And in this particular study here, which is published, you can see what we did is profile the difference in growth of a population in a xenograft mouse versus in vitro in a plate, and we got a very distinct difference where you can see that about only 7% of the original clones that were injected into the mouse actually went to form the bulk of the tumor. Almost 90% of the tumor, over 80% of the tumor was formed by those. So this is, a kind of a straightforward approach. This was done a few years ago. There's more sophisticated versions of these type of studies. This is just published earlier this year by MD Anderson, and this is looking at a PDX model where they've taken cells from the patient, transplanted them in the mouse, and then took them out of the mouse and cultured them, and they found that after they labeled them, transduced them with the library, and passaged for several passages, they went out quite a few passages, they got a core group of cells with a specific set of barcodes that continued to propagate, and they continued to propagate indefinitely, and they put them in the mouse and they continued to propagate. And so using this sort of tumor initiation cells, they were able to then do some studies where they looked at how the population changes when they treat it with different drugs, and they could find that, for example, the population obviously decreases and could be perturbed with certain drug treatments, but certain core groups of barcoded cells in this particular selected population came back regularly each time, and it was a different core group with different drugs, so they were able to actually look at that. And so with that, I could talk a little bit more about some other applications of barcodes, but I'd like to introduce my colleague, Dr. Ian Winters, and he is a researcher from Stanford University, and he's actually done some work, he and his lab have done some work developing an innovative application for the barcodes that combines barcoding technology with CRISPR technology. And so he'll tell you a little bit about that now.